Excellent. Okay. So we're going to be finishing up on photosynthesis. And at the end of this, you will have a kind of detailed kind of story of the evolution of the light reactions in particular. So remember, this is kind of entitled Photosynthesis I. And so photosynthesis I then is only going to be representing kind of the first component of photosynthesis, in particular the light reactions. And so this then will kind of give us not only a baseline for understanding the light reactions of photosynthesis, but we're going to be continuing on with photosynthesis a little bit more kind of mid-semester then, okay? And we're going to kind of interrupt kind of our more formal understanding of photosynthesis because in the story of the evolution of the land plants, okay, the light reactions perform kind of the fundamentals to understanding the clade then that ultimately then gave rise to another subclade known as the land plants, okay? And so we're going to hopefully kind of wrap this into our understanding of the land plant phylogeny. And if we have time today, we're going to talk a little bit more about the major land plant clades. And so kind of depending then on time, we're kind of working up then to our understanding of the invasion of land. And plants are actually quite important because they were one of the first major clades and organisms to actually go from life in the water, an aquatic lifestyle, to actually living on land. And so we have to understand then the really unique selective pressures then for life on land, right? So what then were the selective pressures then that effectively selected then for a lifestyle on land, okay? And so that's what we're going to be working up then towards today. All right, so let's kind of finish up and wrap up the light reactions. And this is going to be important because we need to kind of put these selective pressures for life on land into context, okay? And so we need to understand some basics. And so what I want to kind of go back to our review on plant cells. And so we covered this, yesterday, and we'll be going into more detail on other aspects of plant cells. But for today, what we want to focus on is this organelle known as the chloroplast. And so also remember that there are several different organelles within animal and plant cells. In particular, both plants and animals have both mitochondria and chloroplasts. But plants then are unique, and I just want to make sure that I emphasize this, in that plants then have chloroplasts. Animals in general do not. Okay. So we're going to then be zooming in on the light reactions in particular within the chloroplasts. In particular, we're going to be focusing in on these unique membranes within then the chloroplast itself. So remember, the chloroplast is it has a double kind of phospholipid bilayer on the outside, but then on the inside there are several more phospholipid layers. And within those layers then contain then not only several unique molecules, but also several important proteins as well as enzymes associated then with photosynthesis. And so you're going to hear a bunch of different terms, okay, that can be confusing. And I don't want you to be confused then about these different terms, because within the chloroplast, we're going to be talking about thylakoid membranes, okay? And thylakoid membranes are special membranes associated with the chloroplast and that house then a lot of the components then of the light reactions. And so we're going to be kind of zooming in then and giving you a more kind of formal understanding of what happens within thylakoid membranes. And so within kind of typical biology books, they have these, I don't know, kind of these Dumbo-like drawings of the typical chloroplasts, right? But it's actually much more beautiful. The biology that actually happens within chloroplasts and the structures are actually much more beautiful and much more kind of well designed by natural selection. And so I can kind of give you some of these hints by kind of giving you these, our latest kind of animations in terms of what the biology actually looks like. And so the light reactions then of photosynthesis actually occur then within then these membranes inside then the chloroplast, okay? And so in this animation, what we're doing is we're showing you kind of one chloroplast and zooming out, and you can see then a typical plant cell within a leaf kind of packed and full then of these chloroplasts, all right? So typical leaf, but remember the leaf is green because it's reflecting a lot of this sunlight. And then within then that within then that leaf itself, remember that green then is ultimately coming from the within then the chloroplast itself, within these thylakoid membranes. Okay, so these are great little movies, great little animations. I'm going to be kind of highlighting a few of these then as we move on. And if you want to check out a little bit more of these animations, I encourage you to check out this first year biology animated textbook, which is really nice. Okay. All right. So at last lecture, we kind of ended with kind of emphasizing what's universal about photosynthesis. And so I emphasized two kind of universal aspects then of photosynthesis itself. In particular, what is universal about the photosynthetic chemical equation, but also what's universal about the photosynthetic structure, okay? And so let me just quit out just really quickly here. I just want to make sure there's no one waiting at the door. All right, there isn't. Fantastic. The Zoom door, that is. Sometimes when I'm lecturing, it's easy to forget people. All right. So I emphasized what was universal about the chemical equation of photosynthesis, but also what is universal about the structure of the photosynthetic apparatus itself, okay? And so what's universal about the photosynthetic apparatus itself is actually three things, okay? And I would like you to remember these three things. The first is the photosynthetic structure is made up of these chlorophyll-based light harvesting pigments, okay? And there's going to be some different flavors then of these, all right? And if you go into biology, in particular within microbiology and other aspects of detailed inner workings of photosynthesis, you'll actually find that there are many different types of these chlorophyll-based harvesting pigments. The second aspect of what's universal about the photosynthetic structure is not just the chlorophyll-based pigments, but then there's going to be this structure known as an antenna reaction center design, okay? And it's a physical structure then that's going to be made up of many different components, and it's actually quite complicated. And it is an antenna. It's a highly tuned antenna to then harvest then energy from the sun. And within then that antenna, there's going to be something known as a heterodimeric protein core associated with something known as the reaction center, okay? And all of these kind of structures, these first three structures, this chlorophyll-based pigment, this antenna reaction design center, and this heterodimeric core of this reaction center is all going to be structured all together and kind of honed by natural selection then to harvest energy then from the sun. So it's a physical structure, and it's actually really, really cool. And it only has been within the last, say, decade or so that we've really kind of figured out what this structure really looks like. Okay, so first stop, the first kind of universal aspect of photosynthesis, these chlorophyll-based pigments, okay? And chlorophyll is actually a complicated molecule in and of itself. And what chlorophyll does is actually harvest light by these kind of internal kind of bond reconfigurations then of the chlorophyll molecule itself. In particular, if you remember back, maybe if you've had organic chemistry or biochemistry, you may remember these trans-cis interconversions. Don't worry about those details, okay? But this is a point of departure then for these other courses. And so we're then going to be harvesting then this light by this change then of these internal kind of molecular bonds then of the chlorophyll structure itself to then kind of elevate then the molecule to these greater energy states. And so all oxygen evolving photosynthetic groups, okay, so all organisms then on the planet then that release then oxygen as a part then of photosynthesis use one flavor of chlorophyll, and that is chlorophyll A. But now other photosynthetic organisms on the planet, such as, for example, a lot of photosynthetic bacteria, actually use these other flavors of chlorophyll known as bacteriochlorophylls, okay? So this is important because I'm going to be asking you to define what a plant is. And one definition then of a plant is associated then with whether or not it has chlorophyll A or not, okay? And we'll come back to this, all right? Now, there's going to be different pigments, okay? Within within the land plants, but also in different organisms that do photosynthesis. And those different pigments then actually produce some of the different colors then that you see, not only within plants, but other photosynthetic organisms, such as brown, red, purple, green. And that all depends on which wavelengths then of light is then being harvested then for energy. Now, within the land plants, as well as within photosynthetic algae and other photosynthetic groups, we also have some additional pigments, things known as carotenoids, bilin pigments, and so on. I don't really have too much more to say about that, but they will kind of show up from time to time. But these are all chlorophyll-based then pigments. Okay. Now, I would like you to remember exactly the basic structure of chlorophyll. You should be able to draw this out. I'm kidding. Okay, trying to keep you on your toes here. It's a complicated molecule, okay? This is the basic structure of chlorophyll. And in higher plants and algae and land plants, okay, they have two different flavors of chlorophyll, okay? In the higher plants, the land plants, and in algae, okay, they have both chlorophyll A and chlorophyll B. The difference between chlorophyll A and chlorophyll B is this little kind of modification of this N side group here. Don't worry so much about this. I don't expect you really to remember this. But what I would like you to point out is that we have this long carbon tail. And then attached to that carbon tail, we have this more elaborated series of rings associated with a few different elements, atomic elements that you likely know of, magnesium and nitrogen. Okay. Now this is important because when you fertilize your plants, nitrogen is important, but also you give plants in the normal fertilizer a little bit of magnesium too. And one of the reasons for that is that nitrogen in particular is heavily associated then with photosynthesis. And here's one of the molecules that contains actually a decent amount of nitrogen, chlorophyll. Okay, now notice this is also a ring-like structure. We have this magnesium, nitrogen, carbon are all kind of linked together in a series of these different kind of bonds then that forms then these different rings. Now this is actually important then for basically harvesting then this electron, okay? And so these electrons then are going to be moving kind of freely then within these rings, and that's going to be important. So this is just one chlorophyll molecule. Now, within then these photosynthetic units that we're going to be talking about, within these photosynthetic antennae, you're going to have many different chlorophylls all together, all working together to harvest then the sun. Okay. So here are the chlorophyll absorbance spectra for both chlorophyll A and chlorophyll B. So within then most plants, we're going to be having both chlorophyll A and chlorophyll B. And notice chlorophyll A and chlorophyll B are then going to then be harvesting different wavelengths, okay? So what's great about having chlorophyll A and chlorophyll B is that we can then absorb, all right, different components then of sunlight, okay? Now there are a few other pigments sometimes involved, in particular lutein and beta carotene. They tend to kick in at different wavelengths in terms of their importance. Okay, so one universal aspect then of the structure of the photosynthetic apparatus is then chlorophyll-based pigments. The other is something known as the antenna reaction design center. And what's unique about this, I'm highlighting design here. So it's this antenna and this reaction center, and it's their physical design. And it's actually a unique spatial arrangement between the light harvesting pigments and the reaction then catalyst then within the reaction center itself. So this is a physical kind of spatial design, okay? And this is going to make a little bit more sense as I move on here. Now, in kind of the old days in biology books, they kind of described it like this, okay? We have several different pigment molecules, chlorophyll, these chlorophyll-based molecules. And these chlorophyll molecules are going to be kind of spatially kind of clustered together. And this light, in particular, protons are going to be coming down. And these these chlorophyll molecules are going to be excited, right? So that light then is going to excite then electrons within these bonds, right? And that's going to ultimately pass along then an electron to then this reaction center. So we're going to have this antenna component and then a reaction center. And then that electron, as it hits then the reaction center, we're then going to be interacting then with the donor molecule and another molecule that's going to be acceptor, where then this electron is then going to be kind of stolen and then kind of passed along, okay? So here within this reaction center, we're going to have these protein molecules that are going to be accepting and donating then electrons. So we're going to have this kind of excitation and transfer, and then we're going to be moving then this electron as we transfer them on. So we have this kind of antenna and then this reaction center, okay? So that was kind of the old days, how we kind of, you know, understood things. But now as we kind of move forward, it actually was in 1930 that two scientists named Emerson and Arnold figured out that it actually took the cooperation of about 2,400 chlorophyll molecules to produce one molecule of oxygen. Whoa, that was pretty amazing. When they figured that out, when they actually went through the calculations, they realized that actually you need about a little over 2,000 chlorophyll molecules in order then to actually produce one molecule of oxygen. So I remember 1930, it wasn't that long ago. And so our understanding of photosynthesis was that, well, actually you need this cooperation of chlorophyll molecules, right? All kind of together. And that was our first kind of realization that, oh gosh, these things have to be spatially arranged in a way in order then to harvest light, okay? And so we have this antenna complex of a lot, 2,400 then chlorophyll molecules that are going to be harvesting light energy. But then the other purpose of this antenna complex is then we're going to channel then all of that energy, okay, in terms of an excited electron into something known as the reaction center, okay. All right, and so this was kind of like an updated then drawing, right? So here we have what Emerson and Arnold then discovered in 1930, that we have 2,400 of these individual chlorophyll molecules. And they're all going to be working then together to then get kind of excited in the presence of a proton and light, and then pass along and aim then this electron to this specialized reaction center where we have our electron donor, which is water. And that electron then is going to be ripped off of then water and then passed along to an electron acceptor complex and to a bunch of really cool biochemistry, in particular what we call photochemistry, associated with this electron transport chain that we'll get to in a little bit. So this is the antenna, and here's our reaction center, all right? And so in this reaction center, we're gonna have a special pair of chlorophyll molecules, then that are gonna be transferring this excited then electron, all right, to an electron acceptor then molecule, okay? And it's actually water then that's gonna then be donating then ultimately this electron to all these excited then electrons, right? So I can kind of give you a more up-to-date kind of version of this really cool animation. We're now, thanks to a lot of computational kind of bioinformaticians and biophysicists and molecular biologists, they've been able to kind of put together, and we can zoom in on one of these kind of groups then of the specialized antenna reaction center. And we call this a photosystem. And in particular, this is photosystem 1. All right, so I can kind of show you then this animation of all of these different kind of chlorophyll molecules. And so remember, these chlorophyll and molecules are all kind of packed then within then the membranes, then the chloroplast within these grana. And then they come in these specialized little units, these photosystem units. And you can see kind of all of these chlorophyll molecules all packed together, kind of centered around then a reaction center, okay? And so we can kind of delve into a little bit more detail about how exactly this looks, okay? So it looks kind of chaotic, but this is really, really cool. And if you actually zoom in on one then of these photosynthetic units, one of these photosystems, this is actually what it looks like. This is the photosynthetic unit that consists then of several of these light harvesting units consisting of many of these different chlorophyll molecules. So remember, in total, we have 2,400 chlorophyll molecules, all interacting spatially, and they're spatially arranged around, if you notice this unit here, this is called RC, this is the reaction center, okay? And really what this is an antenna. So you can kind of think of this as like maybe like a radar dish or something, but this is actually a physical antenna that's finely tuned to aim at the sun and these chlorophyll molecules in the presence of a photon will then get their electrons excited in those different rings, all those different rings then with this magnesium and nitrogen, and those electrons are basically going to swirl around and get excited, and they're aimed at the reaction center. And that energy in the form of this excited electron is then going to be aimed then at this water molecule in the reaction center. And there we're going to rip then off and steal an electron from water. That's then going to go on then to fuel, okay? And so again, that energy from the sun is going to excite then electrons, all right, within then this individual unit, this photosynthetic unit, in order then to steal then electrons then from water. And so this is a spatial organization. And that's what's really cool. All right, so we can kind of give a better animation. So what I kind of like about this is that you can see both the red, right, and the bluish light, okay? Remember, that's the light then that these photosynthetic units then are going to be harvesting. And so it kind of looks like these are little lasers kind of coming in. All right, so here's the photosynthetic unit, right? So this is photosystem 1. And you can see both, so that the red and blue light, right, that's getting excited and kind of moving electrons around, right? And it's all aimed, that photosynthetic unit, photosystem 1, all of that energy is then aimed then at the reaction center right in the middle, right? And so then that's where water is then going to be losing then its electron. And then that electron is then going to be used in the electron transport chain, okay? So what you're seeing, each of those little blue and red flashes, that's a photon then associated more with blue light or red light, right? And so each of these photosynthetic units are then harvesting then that energy, making use of that energy to steal electrons. Okay, awesome. All right, so what's really cool about evolution by natural selection, is if you remember back, I kind of taught you a little bit about, we could talk in terms of how traits evolve, how phenotypes evolve, and we can talk about evolutionary convergence as well as evolutionary divergence, right? And so across all photosynthetic organisms, we see the evolution then of the light reactions. And we thought that it had occurred, it only evolved one time on Earth, okay? The light reactions. And in general, that's true. But this is what's really great about being a microbiologist, is that there's so many microbes on Earth and so many really cool biochemistry and cool aspects of their biology to study. And so there's a group of bacteria, it actually turns out. These bacteria are known as halobacteria. And it looks like we have not one, but two independent evolution events of the light reactions, in particular, the light reaction structure, okay? And so if you've ever been in the Owens Valley of California, this is on the backside of the Sierras in California, sometimes you'll come across, this is a really arid environment, right? Really desert-like environment. And sometimes you'll come across then these lakes, okay? These arid then lakes. And within then those lakes, sometimes when it starts to get really hot, they'll start to turn pink, right? And so what's kind of cool then about these lakes turning pink is that that's actually a big bacterial bloom, in particular these halobacteria. And they tend to occur in these extremely saline or salty environments. And what's cool about halobacteria is that they perform photosynthesis. But when people started to look in more detail about kind of the structure and looking at the structure, they noticed that there was a lot of similarities in the light reactions of halobacteria, but there was one important deviation. And that is they contain actually, they don't necessarily contain chlorophyll, but instead they contain this retinol protein system. So you may have heard of retina, your retina, which is your eye. And this protein, retinol, is very sensitive to light. In fact, it's effectively the same protein that you have in your body, in the back of your eye, that's very sensitive to light. But what it looks like is that halobacteria independently evolved another set of light reactions very similar to what we see in the light reactions then of other bacteria and within the land plants and algae. But what's interesting is that halobacterian people actually have a common type of membrane protein, these retinal proteins. Now, it's not that we actually shared a common ancestor with halobacteria then that gave rise then to both halobacteria and us, that contain then these proteins, but it looks as if this is an independent evolution of this retinal protein within halobacteria. And this is what is actually activated then by sunlight. And so that's what's really cool about the photosynthetic structure then, is that we have the independent evolution of this same antenna reaction design complex, right? It's the same solution to harvesting sunlight, but it's done just totally differently. It's built with different parts. And within then the center, all right, we also see these bacterial rhodopsin. Okay, this is the retinal protein then that is very sensitive then to sunlight within halobacteria. So that's cool. So the consequence of this is that it looks as if photosynthesis on Earth has likely evolved at least twice, 2 times independently. And the biochemical evolutionary kind of implications is that we see a convergence of these light reactions, but they're kind of built then from different parts, in particular bacteriorhodopsin instead of chlorophyll. Okay, so that's pretty cool. Photosynthesis has likely evolved at least twice. All right, so the third universal then aspect of the structural components of photosynthesis is something known as the dimeric protein complex, and this is the reaction center, okay? And in this reaction center, this is where we're converting all that energy from the sun to a usable form. And it turns out that there's 2 types of reaction center. And it's not important for you to really know, I don't want you to memorize these 2, but you should know them when you see them, okay? These phenophyton and quinone type, and an iron sulfur cluster. These are 2 types then of reaction centers, kind of 2 different flavors that we find then within plants. And from a variety of different groups and clades, okay? But within cyanobacteria and then within eukaryotic photosynthetic organisms, both of these 2 different types coexist in the same organism. And that's actually very important because these 2 different types of reaction centers are known as photosystems, photosystem 1 and photosystem 2. And what's interesting about photosystem 1 and photosystem 2 is that they're associated then with the different wavelengths of light then that you harvest. Okay, and so if you remember, plants then are actually kind of interesting because they actually reflect then green light. That's why plants are green. And so photosystem 1 and photosystem 2 are associated then with harvesting the different components then of sunlight, in particular, more of the blue light and then more of the red light. Okay, so what are these photosystems? All right, so I showed you in those animations, these photosynthetic units. That's a photosystem. And so these photosystems are clusters then of light absorbing pigments, and they're associated electron carriers in the membranes of chloroplasts. It's the photosynthetic unit. It's all the chlorophyll molecules aimed within this antenna and the reaction then center itself. And again, they're forming these clusters known as photosystems, but there's 2 types of photosystems based on these differences then in the electron reaction center. Okay. These photosystems then absorb light over different spectrums of the visible spectrum itself. Okay. So in higher plants, in particular within the land plants and some green algae, we have these coexisting reaction centers. And so photosystem 1 and photosystem 2 together allows for more complicated functions when you put them together. In particular, we have more efficient photoautotrophy, better ways of harvesting then energy from the sunlight. And so we can then absorb energy then from different wavelengths in order then to minimize fluctuation in energy supply. So remember that video from the last lecture? This is where this comes in. So these coexisting reaction centers are together going to be stealing electrons, right? They're going to be trapping light energy, and they're going to be creating a proton gradient then to generate ATP. And in the process, we're also going to be producing high energy compounds. Okay, this is all about ultimately kind of stealing energy from the sun. And there's a few different ways by which we can do that. Now, in your introductory biology class, you likely kind of saw then these different kind of these different drawings, right? Where we have the sun, right? We have these photons coming in. We have these different chlorophyll then based molecules, chlorophyll A, chlorophyll B. We also have keratinine and xanthopills that are kind of harvesting additional wavelengths of light. We're now aiming them then at the reaction center, and we're passing them that along to these electron transport chain. Okay, so here's our chlorophyll-based pigments, here's our antenna reaction center design, and then here's our reaction center. These are the three main components of what's universal in then the photosynthetic light harvesting structure itself. All right, but remember we have these different photosystems, okay? Each of these different photosynthetic units where we have our chlorophyll-based molecules, okay? We're having our reaction center design, right? But now we're going to be putting then these two different reaction centers and photosystems then together. And so it actually turns out, it's a long story, don't worry about it, but photosystem 2 starts first, and then we have photosystem 1. That's just how they're labeled. And so photosystem 2 harvests light in wavelengths then that are less than 680 nanometers, whereas photosystem 1 is going to be harvesting light then that's longer than 680 nanometers. Okay, and they're going to be coordinating with each other and working together in that we're going to be sharing then a stolen electron, and we're going to be passing electrons then along, ultimately to create then a series of different molecules and stored energy that ultimately is going to go to something known as the Calvin cycle. These are the carbon then reactions, which we're going to be talking about later on in class. So you likely have had bits of this then from your introductory biology in terms of these different photosystems. But I just want to emphasize that these two different photosystems are then going to be differentially harvesting then different wavelengths of light. Okay, so we can kind of distill all of this in terms of these cooperating photosystems, photosystem 2 and photosystem 1, in terms of this electron transport chain. And so here, what I'm showing you here are these different photosystems, photosystem 2 and photosystem 1. And together, they're going to be operating in this coordinated fashion. And the end result then is kind of increasing the potential then energy and the stored energy then of the entire system itself, right? So we're going to be harvesting then light energy, kicking up our potential then for energy then storage in the system, and passing then that stored energy along in terms of kind of the stored energy itself and a DPH. So there are three main functions then of the light reactions, okay? So remember, we have the universal set of equations then for what's universal about photosynthesis, what's universal about the structure of photosynthesis, and the end result of that are the three main functions then of the light reactions. And that is to steal electrons, to capture that light energy, and to store then that captured energy in the form then of these stored molecules. Okay, so these are producing these high energy then molecules then that we're going to then use to fuel the rest of metabolism. Okay, and so this is sometimes called the Z scheme of the electron transport chain because it looks like a Z kind of on the side, right? The Z scheme or the electron transport chain. All right, don't worry so much about the details, but you may have seen this in your introductory biology. Okay, so then let's kind of back up then and put this into a little bit more kind of physical kind of construct. Here then is one of these thylakoid membranes within the chloroplast. This is a phospholipid bilayer. And kind of floating then in these membranes are these different photosystems. Here's photosystem 2 and photosystem 1, as well as several different proteins and membranes that are all going to be operating together. And so this is another kind of photosynthetic unit, okay? Not only do we have our independent photosystem units, but together we're going to couple then that with this electron transport chain, which consists of several different molecules. For this class, I don't expect you to memorize these different components, okay? That's for a whole nother class if you're interested. But there are several different kind of proteins and associated enzymes then that we're going to be linking them together into this electron transport chain. And so again, what's going to be happening then here, think of the overall functioning then of all of these units then together, okay? We're going to be harvesting light in both photosystem 2 and photosystem 1. We're then going to be stealing electrons, okay, within these reaction centers, where water, in this case, is then going to be the source then of this electron then that's stolen. In the process, oxygen is released. That electron is then passed along, and the energy state in total then of the system is kicked up by the operation of both photosystem 2 and photosystem 1. And in that process then of that stolen electron is then used to take NADP and to store then that electron in terms of NADPH. So remember, this is an oxidation reduction process. Harvesting light, stealing electrons, and then we're creating these high energy containing molecules, NADPH. Okay. So we're also at the same time generating at each of these different steps, we're then producing protons. Okay. So not only is an electron then stolen, but from water, but we're also releasing protons, not only from the cleaving then of oxygen and the stealing then of this electron, but also protons then are passed along at different points. And because of which, notice these protons are then kind of dumped within the lumen, the inside then of one of these kind of envelopes then of the chloroplast. But then the outside of the chloroplast, the stroma, notice that in this process, we're also taking protons from the outside and dumping them on the inside. And the end result is we're generating a proton gradient, okay? We're harvesting light, stealing electrons, and we're generating a proton gradient between the outside and the inside of the thylakoid membrane. And that's important because ATP synthetase is sitting right here, and we're using that proton gradient then to generate, we're taking ADP to produce ATP, okay? So we're creating energy storage molecules of ATP and NADPH. Okay, so you should know the main functions then of the light reactions. And so the main functions of the light reactions are to harvest light, to steal electrons, we're generating a proton gradient, and we're using the stolen electron and this proton gradient to create this energy, these energy storage molecules, ATP and NADPH, okay? Now, I can try to summarize this in our animations, okay? So again, here we have, this is the inside of a chloroplast. Here are these stacks then of thylakoid membranes within then the chloroplast. And so we'll play this here. And so we're gonna zoom in, and you can see then these individual photosystems, right? And these photosystems are then kind of linked, and you can even see ATP synthetase there, right? And so this is then our electron transport chain, also coupled with ATP synthetase, okay? And so that's pretty cool. So all then of these different photosystems and these different molecules and enzymes are kind of floating then and kind of embedded then within these phospholipid bilayers. And so you can see the outside and the inside, this is where you're gonna be creating this proton gradient. And so that ultimately ATP synthetase, you can see it kind of all the way at the end, we're actually producing ATP. That's it's kind of being shot out right there, okay? So all of then of these different complexes are working then together. All right. So if you have any kind of detailed questions or want to see these animations, I would point you to the link, you know, for a more kind of full, kind of formal set of animations of these, which are pretty fun. All right. So let's then kind of end up in terms of where kind of the ATP and the NADPH goes from the light reactions. And we're going to be kind of jumping kind of off from here because this involves going into more detail about the Calvin cycles. And sometimes we call these the light independent reactions because they don't involve light or light energy. These are also known as the enzymatic or the carboxylation series of reactions. All right, so sometimes we have these different terms, you know, for this other kind of set of biochemistry, the Calvin cycle or the light independent reactions then of photosynthesis. And here we're going to be dealing with something known as carboxylation, which involves a very important enzyme. And this is also going to be requiring energy, okay? And so we're going to be doing a lot of reducing in the Calvin cycle. And so this is energy dependent. And that's where this ATP and NADPH is going to be important. because we're going to be utilizing the stored energy to fuel the Calvin cycle. And this is also going to be a regenerating process. We're also going to be making kind of a lot of the basic components involved in the light reactions. And this is also going to be utilizing a lot of energy as well. And so this turns out that if we kind of step back and kind of think about photosynthesis in general, that plants have in general plenty of light. There are environments where light may be limiting, but most of the time, what actually influences variability in photosynthesis, it turns out, is not so much the light reactions in terms of the presence of ATP and NADPH, but instead, what gets really tricky for plants is actually the Calvin cycle. And so what regulates kind of photosynthesis in terms of variability of photosynthesis oftentimes turns out to be the carboxylation reactions. And so we'll have much more to say about that. So that's the topic of kind of future discussions and lectures. And we're going to be putting then kind of all of these together. So for right now, it's important for you to understand, some of the big pictures of the light reactions and what's universal about photosynthesis, but also these differences in photosynthesis between photosynthetic bacteria and photosynthesis then within the land plants and the higher plants. And so the light reactions are a source then of electrons and protons. And so remember, I gave you some examples of using either hydrogen sulfide or water, okay? And so we're dealing with light and pigments and electronic, the transfer then of energy through electrons, okay? And so these are all kind of pieced together via the electron carrier train, but then we also have these waste products. And so I give you one example of a waste product, and that was sulfur, okay, sulfur producing bacteria. But then within a lot of the higher plants, the waste product here is oxygen, right? Now we're going to be interacting with a lot of different substrates as well. And I gave you kind of an overview of that earlier in photosynthetic bacteria, as well as within algae and a lot of the higher plants. So some of these substrates then that we're going to be dealing with is either CO2 or nitrogen or nitrate or some of these various different nitrogen compounds then that are involved in ultimately the second round then of biochemistries kind of associated with ultimately fueling the growth of photosynthetic organisms, okay? So we've only focused on light reactions. We're gonna come back and put all of this together in terms of the carbon reactions later, okay? All right, now let's kind of step back and kind of put all of this, into focus. And here, this is a kind of a busy little graphic here, but I would like you to kind of we'll step back and we'll walk through this. And so the main thing I want to point out here is that we're putting kind of all of this into the context then of life on Earth. And so here I'm showing you kind of the age of the Earth going from about 5 billion years all the way then to present day, okay? And there's going to be quite a few kind of mileposts then that's kind of happening kind of along here. And we're going to walk along this here, all right? So if you remember, we talked about the biological evolution of several kind of key kind of components. And I do want you to memorize these dates, okay? And I've already emphasized a few of these key dates, in particular the evolution of these protocellular organisms, okay, approximately 4.5 billion years ago. But we also get the evolution of photosynthetic bacteria approximately 3 billion years ago, okay? And then we find evidence for the evolution of oxygenetic photosynthesis. That is photosynthesis then that's producing then oxygen as a waste product. That comes along approximately 2.5 billion years ago. Now, remember, it wasn't until approximately 1.5 billion years ago that we get the evolution of eukaryotes and algae. But then what we call the higher plants or the land plants is occurring sometime around approximately 500 million years ago. And we're going to hone in on this date a little bit later on. Okay, so that is our crude kind of timeline going from protocellular organisms to the evolution of oxygenetic photosynthesis, then to the evolution then of the land plants. And I want to emphasize one important implication for the evolution of photosynthesis on Earth. So if you notice, as we go back to about 4 billion years ago, and we look at the concentration of CO2 in the atmosphere, the concentration of CO2 in the atmosphere was exceptionally high. Most of the atmosphere, well, I wouldn't say most, but a large concentration then of gas then within the atmosphere of Earth before the evolution of photosynthetic bacteria. There's a lot of CO2 in the atmosphere and relatively small amounts of oxygen. But once we get the evolution then of oxygenic bacteria, in particular the photosynthetic bacteria and the oxygenetic photosynthesis, as soon as we see the evolution of oxygenetic photosynthesis, the concentrations of oxygen within then the atmosphere start to rise. But also notice with the evolution then of photosynthetic bacteria, we also start to see the reduction in terms of the CO2 within the atmosphere. And this is really important. So one question I will likely ask you is, what's driving the reduction then of CO2 within the atmosphere? Now, the answer to that is actually a little complicated because it also involves geology and rocks and what rocks then are present. But another important driver of the reduction of CO2 in the atmosphere is the evolution of photosynthesis. So the evolution of photosynthetic life actually has started to scrub the atmosphere of CO2. And the evolution of oxygenetic photosynthesis has also led to the the increase then of oxygen within the atmosphere. In terms of the evolution of life on Earth and the implications then for the geochemistry and biogeochemistry on Earth, the evolution of photosynthesis was huge because it helped shift one of the kind of major components of the atmosphere, CO2, kind of shifting then the atmosphere from more CO2 dominated to more oxygen dominated. Okay, and that had very important implications. And in terms of the relative contribution of photosynthesis on life on Earth, earlier, around 3 billion years, it was mainly photosynthetic bacteria. But then that shifted to a dominance of blue-green algae, and then that shifted to eukaryotic algae, and now most of the photosynthesis on the planet is dominated then by what we call the higher plants, in particular, green algae and the land plants, okay? So the evolution of photosynthesis is extremely important for the atmospheric chemistry of life on Earth. Okay. So I'll kind of skip over those questions because we focused on those last time, but that's for your own edification. Those questions will come up in our next quiz and midterm exam. So here, what I would like you to do is, it'd be great if someone can give me a definition of a plant and kind of define what a plant is. And it'd be great if someone wants to either mute yourself. We'll take a few different answers. And I think I'll take 4 different answers before we move on. So it would behoove yourself to at least give me a definition. Can anyone give me a definition of what a plant is?
Something that survives from sunlight.
All right, something that survives on sunlight. That's one. Thank you for that answer. All right. I want 4 different responses.
An organism that contains chlorophyll A.
Ah, okay. All right, I like all of these, all right, because you're getting close. You're getting really close. You're getting really hot there, all right? I like that. An organism that contains chlorophyll A. I want another answer.
It also contains chlorophyll B.
Ah, also you're getting really, in fact, I really like that definition much better. An organism that contains chlorophyll A and chlorophyll B. Is there another? You could also say, I agree with one of the answers.
I would agree with those last 2 things.
Oh,
sure.
Okay. All right. All right. So I'll give you a big hint. When I ask you to give a definition then of either like a plant or a given taxonomic group, I want you to think phylogenetically. And I want you to think in terms of synapomorphies and clades, okay? Because that's how we're gonna define then biology, basically. We're gonna define in terms of clades. And so a plant then, it's an interesting term, what is a plant, right? Because it brings different connotations. But we have to do this phylogenetically, and we have to do it in terms of synapomorphies. Synapomorphies then will define the clade. And so the answer, chlorophyll A and B, is a definition of a plant. Okay, that is one definition of a plant because then that characterizes a clade. Okay, you could also characterize it maybe as, well, if you said photosynthesis, okay, one of the answers was, well, it's an organism that performs photosynthesis. Well, that's interesting because that's not necessarily a clade because there are some photosynthetic bacteria as well as the land plants and photosynthetic algae, and together they would not form a clade. Okay, so remember these differences between paraphyletic groups and so on, right? All right, so I'm going to ask you this question later on, okay, in particular during a quiz and your midterm, all right? So that's good because then that's going to lead us into this next kind of part because we want to be able then to define then all these early events in plant evolution. And so I'm going to come back then to that definition of a plant. And I like the definition contains chlorophyll A and B because indeed that is a synapomorphy of one of the ancestors then that gave rise to one of the clades. Okay. Now, what is truly remarkable about Darwin's insight is that all of life ultimately has one common ancestor. Okay. And all the embryophytes, the land plants, also had one common ancestor, then that gave rise then to the descendants then of all then taxa then that are the embryophytes or the land plants. So the embryophytes have their origins in the ocean, aquatic environments, and they're ultimately related to eukaryotic multicellular algae. Okay. And so I'm kind of cutting to the chase here. And so we find then the common ancestor of the land plants in terms of a relationship with eukaryotic multicellular algae. So it turns out that photosynthetic prokaryotic unicellular bacteria, within then these prokaryotic unicellular bacteria, we have groups of organisms, okay, that we can trace to an ancestral eukaryotic photosynthetic organism. And in particular, within the green algae, all green algae are photosynthetic and they have chlorophyll A and B. These are multicellular algae. And within then the green algae, we find a group of green algae that are most closely related to the land plants. Okay. So we know based on kind of morphology and based on then genetic relatedness and kind of peering into their genomes, that land plants are ultimately kind of linked and most closely related to a group of plants known as the green algae. And green algae have chlorophyll A and B, okay? So let's try to piece together these different timelines, all right? And so I'm showing you here, this is a little complicated, but I'm gonna try to walk you through this. And so this is the geologic time chart, okay? And so I don't expect you to memorize all these different components, right? All these different names, but you should know approximately kind of where you are in the grand scheme of things, right? So BYR here is billions of years, right? So we're going back to the Precambrian, about 4.5 billion years ago. And we're kind of piecing together all the geologic time charts. And so I expect you to know the times of the first radiations of the major plant clades and groups then that we cover in class. And so the land plants or the embryophytes appear to our best kind of fossils and our best molecular dating to have originated somewhere coming out of the Ordovician into the Silurian. So approximately 450 million years ago, and definitely by 400 million years ago, we see very clear fossil record and molecular evidence for the evolution of the land plants. Okay, so somewhere here, plants then went from being mainly aquatic, okay, within the ocean, to then also occurring on land. All right, so this is a big transition event, right? So what really was it that actually pushed then plants, selected then for specific populations to stick their little green heads above then the water and start then to invade the land? Okay, so the Silurian, right? This is a very important time period in terms of plant evolution. Okay, so around between 400 and about 450 million years ago, that's when we first start seeing evidence then from the embryophytes. All right, so let's try to piece kind of all of this kind of together. All right, so I'm showing you here a phylogeny of the eukaryotes, and I don't expect you to kind of memorize this at all, but we are going to zoom in on kind of one branch then of the eukaryotes. And I also want to note this big polytomy here. And there's been a lot of work kind of piecing out kind of this from this ancestral eukaryote then that ultimately gave rise to animals and fungi and red algae and the plants and other protists and everything. A lot of very important evolution. So there's really cool work kind of piecing together all the different steps right here. But we're just going to show it as this big explosive radiation here in terms of this polytomy. So we can zoom in then to one grouping then of the eukaryotes. In fact, one clade called the green plants, okay? And so we're going to define then plants based on a group of plants known as the Viridiidae plantae, the green plants. And so the green plants then consist of the green algae and the land plants. So notice not all algae then are within this group, but within the green plants consist of green algae and the land plants, okay? And so we're going to zoom in then on this group. And so here's a very simplified phylogeny of the Viridiidae plantae, or the green plants, okay? Now this has been worked on quite a bit, so this is a very simplified phylogeny. I don't expect you to memorize this phylogeny, okay? But you should know it when you see it, okay? And so our best evidence suggests that around 800 million years ago, the green plants then evolved. All right, so remember, this is coming then out of the eukaryotes, okay? So there was an ancestral eukaryote then that gave rise then to the green plants, consisting of unicellular and multicellular green algae, as well as this one little branch up here, which is the embryophyte, okay? And sometimes we call that the kingdom plantae, the plant kingdom, okay? But we're gonna define plants then as the green plants, okay, that evolved approximately 800 million years ago. And the green plants then consist of unicellular, multicellular green algae, as well as the embryophytes of the land plants. Now, it turns out that this ancestor, this common ancestor that gave rise to this clade, contained then this very unique synapomorphy of chlorophyll A and B. So that is one definition of a plant. One definition of a plant is chlorophyll A and B, and that definition of a plant includes the embryophytes and the unicellular and multicellular green algae, because they're all derived from a common ancestor then that had this one trait, chlorophyll A and B, okay? So again, I don't expect you to memorize the phylogeny of the green plants, but you should know it when you see it, okay? So the green plants evolved about 800 million years ago. All right. And another interesting synapomorphy associated then with the green plants is also a molecule known as cellulose, okay? And we'll talk more about cellulose as we move forward. So chlorophyll A and B, main synapomorphy, but also we start to see a molecule known as cellulose, okay? And we'll come back to both chlorophyll and cellulose later. All right, now I'm really jumping ahead because I'm showing you a phylogeny that I do want you to memorize, okay? And so this is gonna be your main phylogeny that I'm gonna want you to memorize because we're gonna be kind of adding on to this, right? I will ask you on the next quiz to draw not only this phylogeny, but also to label this phylogeny, okay? So I'm giving you a heads up. I expect you to be able to draw this out, okay? So here then is a group, in this phylogeny, we can identify several different groups then of organisms. In particular, a group of organisms known as the chlorophytes and the charophytes. These then make up green algae. Green algae consists of the chlorophytes as well as the charophytes. Charophytes are interesting because they share a common ancestor then that's most closely related then to the land plants. And so it turns out that the land plants, or the embryophytes, consists of these group of plants: the liverworts, hornworts, mosses, and tracheophytes. Oh my gosh, I'm dumping on you all these different names. Now we're gonna spend plenty of time then with these groups of organisms because they're really cool and they're awesome. All right, so you should be able to tell me which group then of plants, which taxa then make up the embryophytes. And you should be able to tell me, well, that's the liverworts, the hornworts, the mosses, and the tracheophytes. Now, it turns out that these also have different names, okay? Liverworts, hornworts, and mosses consist then of the bryophytes. The tracheophytes, well, those are the vascular plants, okay? So we're gonna be elaborating this phylogeny, and we're also gonna be focusing in then on the common ancestors of each of these nodes, and we should be able then to identify the synapomorphies of each of these nodes, because when we identify the synapomorphies, these synapomorphies are associated with the unique evolutionary pressures for life on land, okay? So you already know then the synapomorphy then for one, the common ancestor then for the green algae, bryophytes, and the vascular plants, or the embryophytes altogether. That's chlorophyll A and B, okay? Now, we're gonna be coming back to this phylogeny. Now, I'll also kind of emphasize here that there's also current debate and research to kind of clarify the relative positions of liverworts and hornworts together. And we'll say more about that later. But we're gonna leave that for a later time, okay? And so what I would like to do for this last, I think 10 minutes of class here, is we need to kind of fill in some gaps of what happened in these steps here. Why was it? What were the selective pressures that kind of gave rise then to individuals of green algae then that could not only survive on land, but also reproduce on land? I mean, imagine that. Imagine your green algae in this happy aquatic, nice, tropical, warm water, right? Why the heck would you ever want to live on land, right? What were the selective pressures for being pushed out of the water, at least one group, one lineage, and then living on land? How did all of that work, right? What were the first steps, right? And so we call this the invasion of the land, right? And plants are kind of cool because they were actually one of the first organisms to not only survive on land, but also reproduce on land. So the first organisms to survive and reproduce on land were actually multicellular plants. There's also a debate whether or not fungi were also there too. But it's pretty clear that plants had specific adaptations for growing and successfully reproducing on land, right? So how do we piece together those steps? These were the first organisms that actually poked their little head above water and actually started to live on land. And again, this is important because remember, animals are still in the water, and ultimately plants are providing the shelter and food for the first animals. And again, that's the energetic basis then for the terrestrial ecosystem. All right, so let's piece together what then was this botanical invasion of the land. Well, let's rephrase that question, because it turns out in order to understand how plants were able to invade land, we need to rephrase that, because it wasn't actually an invasion of the land, it was an invasion of the air, going from the aquatic landscape to basically living in the atmosphere, right? So going from water to an air or an atmosphere environment. And actually, this required several key morphological and physiological departures from these ancestral aquatic then taxa and species. So there's a lot of problems because there's problems of desiccation and controlling the loss of water, right? So when you're out, when you go from an aquatic landscape to the land, well, you're gonna be losing water, right? It's hot on land, right? It's drier, obviously. And so the first plants were then actually these land plants that share very close ancestors to mosses, hornworts, and liverworts. Okay, and so we can find some of the answers in terms of studying these common extant kind of taxa. And these guys are really small in stature. In fact, they're really restricted to really wet areas, and we'll have a lot of fun things to say about that. All right, so this was an invasion of a land. And so how do we understand these early selective forces then on land plants? And what we have to do is kind of turn to understanding some of the first principles actually of physics. And these first physics, kind of physical processes help us understand the selective forces operating on the land plants. And so what I would like to do is I want to kind of show you something known as Fick's law, okay? And we're going to start with Fick's law because this enables us to understand what actually selection was operating on those first steps to the invasion of the air. All right, and so I'm actually going to start this.
When molecules or the particles direct the motion of the
Yeah.
particles,
There
the
we
behavior
go.
of the suspension is dictated by random Brownian motion. If you were to track these diffusing particles, on average there is no net motion. Despite this, we know that molecules can diffuse into and out of cells. Motion with a defined direction. So how is this possible? Consider a collection of molecules near the membrane of a cell. Assuming the molecules can freely cross the membrane, some of the molecules will diffuse into and out of the cell. Since diffusion is a random process, the number of molecules leaving the cell will be some percentage of the total number of the molecules inside the cell. The same is true of the molecules on the outside of the cell. Over time, a fraction of these molecules will randomly pass through the membrane into the cell. So, despite the random motion of each molecule, a net inward flow will happen if the concentration outside the cell is greater than inside. We can now define a quantity known as flux, which is represented by the letter J. The flux is defined as the rate of flow, dN dt, per unit area A, across the membrane. The flux will depend on the difference in concentration, delta C, the separation between the two regions, delta X, and the mobility of the molecules is given by the diffusion coefficient, d. If we can determine the flux of molecules and we know the total area of the cell, then we can find the total rate of flow of molecules into the cell.
Oh, that was awesome. That was awesome. Fick's first law. Okay. Now, in this class, whenever I show you an equation, and don't worry, there's not going to be a lot of equations, but whenever I show you an equation, the first thing you should ask yourself, the very first thing is, what can natural selection operate on, right? So you see an equation, now you're going to have a bunch of different variables, okay? So you have notation for different variables, and we're going to ask ourselves, what can natural selection operate on? Okay, so it actually turns out that Fick's first law of diffusion is a fundamental equation shaping how plants work. And Fick's first law is going to come up repeatedly in this class. And it's actually a pretty simple equation, because what we're interested in is starting with a process known as passive diffusion. And I would encourage you to go back to that YouTube video to maybe watch it again, learn a little bit about diffusion. All right. And so in passive diffusion, we're going to have a bunch of molecules of a given flavor. It could be oxygen, it could be, I don't know, sugar or something. And these molecules then, through random kind of Brownian motion, are going to be kind of in the process over time, kind of randomly diffusing, all right, across then a given boundary, a given area, okay? And so we can talk about different solutes, okay? And this is passive diffusion. And that diffusion distance then is x here. So ds dt is the change then in solutes as a function of time. And so this is the rate of diffusion. And we're gonna have a diffusion coefficient then that's gonna be unique to the molecule flavor that we're interested in. So that we could have a diffusion coefficient for CO2, or for sugar, or for nitrogen. It depends on the molecule we're interested in. And that's going to be a constant unique then to that molecule. But then we're going to have this concentration gradient, this delta C over delta X here. So that's the change in concentration. So C here is the change in concentration as you go from the outside then to the inside, right? And so here, the concentration gradient is high, right? Because we have a big then change over that distance, okay? Now, A then is the surface over which diffusion occurs, right? Now, it turns out we can use this equation to tell us why natural selection would favor individuals to live on the land or in the air versus living in the water. Okay, we can actually use this equation to tell us that selection should actually select for some specific individuals. Okay, and so when you see an equation like this, I'll ask you questions like, what if this concentration gradient is large and for a fixed then distance? As this then increases, what happens with the rate of diffusion? Does anyone want to chime in really quickly and say? As this numerator becomes really large, and if delta x is fixed, what happens to the rate of diffusion? Okay, no one's chiming in. All right, so what turns out, as then delta C then increases, the rate of diffusion then increases as well. Okay, so there's also going to be a direct dependence on the rate of diffusion on the surface area, okay, but also the rate of diffusion is going to be inversely then related to the distance, okay. So as the concentration gradient then changes, all right, the rate of diffusion then will also then change, okay. All right, so Fick's law helps us understand the evolution of plant form and function. So this is the last thing I'm going to end with, and so I'll need about maybe 2 more minutes here. And so for a given chemical species I, so this is maybe CO2 or oxygen, right? We can calculate the diffusion rate. And if we know the concentration gradient, CO2 outside versus CO2 then inside the cell, and if we know then the surface area, such as the size of a cell through which the diffusion occurs, and if we know the transport distance, the average distance from the outside then of the cell to the inside of the cell, we can actually calculate the rate at which diffusion will occur, okay? So plant life is actually governed by this very simple process. So think about photosynthesis. We're gonna be assimilating CO2 in order then to grow, but CO2 has to diffuse to the inside of a cell. And so we can ask ourselves, does CO2 diffusion, remember because CO2 is used in the carbon reactions for photosynthesis, does CO2 diffusion happen faster for a cell, photosynthetic cell, inside water or in the air? Okay, we can use Fick's law to actually address that. So let's assume an area. Make it simple, 1 meter, okay? I don't expect you to do this calculation, by the way, but I'm going to use Fick's law then to show you and prove to you that a photosynthetic organism that relies on the diffusion of CO2 is going to perform photosynthesis at a much higher rate in the air than in the water. So we can calculate the diffusion coefficients of CO2 for air and the diffusion concentration coefficient of CO2 in pure water. Notice that these numbers differ. In fact, they differ by a few orders of magnitude. In fact, many orders of magnitude. Huge difference. And if we know the concentration of CO2 in pure water and the CO2 concentration in air in the atmosphere, we can now calculate the rate of diffusion of CO2, because remember, CO2 is important for photosynthesis. This is what plants then, photosynthetic organisms, green plants are using then to grow, right? And so what we can do is we can calculate who has an advantage, a photosynthetic organism that could grow in the air or a photosynthetic organism then that could grow in the water. Well, it actually turns out these are the calculated flux densities. This is the rate at which CO2 is then diffusing. So who has the advantage here? Rates of diffusion of CO2 inside, from the outside to the inside of plant cell is 5, 6, 7, 2 orders of magnitude faster living in the air than living in the water. So if you can poke your photosynthetic organism, if you can poke your head outside, you can diffuse CO2 much faster in the air than in the water. the terrestrial plant will have the advantage. You can complete your life cycle, produce more offspring, and grow at a faster rate if you're performing photosynthesis in the air. The terrestrial plant will have the advantage by 2 orders of magnitude. That's a huge difference. Okay, so we can use Fick's law to tell us who has the advantage. Now, we're going to follow up on this in the next lecture because it turns out that the aquatic plant has the advantage over the terrestrial plant in terms of controlling water loss. So with that, a few minutes over and we'll end right there. So I'll stay on if there are any questions. And with that, we'll see you then next Tuesday. Have a great weekend. Be safe, everybody out there. Please be safe.